बहुत बहुत स्वागत है आपका बायोमोलिक्यूल्स के एनसीईआरटी लाइन बाय लाइन डिस्कशन सीरीज में। चलिए हम लोग डिस्कशन स्टार्ट कर लेते हैं। 9.10 दैट इज मेटाबॉलिक बेसिस फॉर लिविंग। जल्दी से रिवाइज कीजिए। द फर्स्ट क्वेश्चन, dash dash pathway can lead to a more complex structure from a simpler structure. For example, dash dash acid becomes cholesterol or lead to a simpler structure from a complex structure. For example, dash dash lactic acid in our skeletal muscles. The next, the former cases are called dash dash pathway or anabolic pathway. Next, the latter constitute degradation and hence are called dash dash pathways. Next, anabolic pathway as expected consume dash dash. The next, assembly of a dash dash from amino acids requires energy input. The next, on the other hand, dash dash pathway lead to the release of energy. The next, for example, when dash dash is degraded to lactic acid in our skeletal muscle, energy is liberated. The next, this dash dash from glucose to lactic acid which occur in dash dash metabolic step is called glycolysis. The next, living organisms have learned to trap energy liberated during dash dash and store it in the form of chemical bonds. The next, as and when needed, this bond energy is utilized for dash dash osmotic and mechanical work that we perform. The next, the most important form of energy currency in a living system is the bond energy in a chemical called dash dash, that is ATP. The next, how do living organisms derive their energy? The next, what strategies have they evolved? The next, how do they store this energy and in what form? The next, how do they convert this energy into work? The next, you will study and understand all this under a sub-discipline called dash dash later in your higher classes. The next topic in 9.11, the living state. जल्दी से रिवाइज कीजिए इसको. The first question, at this level you must understand that the tens and thousands of chemical compounds in the living organism, otherwise called dash dash or dash dash, are present at concentrations characteristic of each of them. The next, for example, the blood concentration of glucose in a normal healthy individual is dash dash, while that of hormones would be nanogram per ml. The next, the most important fact of dash dash system is that all living organisms exist in a steady state characterized by concentration of each of these biomolecules. Are you agree with it? The next, these biomolecules are in a dash dash flux. The next, any dash dash or dash dash process moves spontaneously to dash dash. लोग जल्दी से रिवाइज नहीं करते यार, जल्दी से रिवाइज करो। नेक्स्ट पोर्शन, चलिए द नेक्स्ट, द स्टडी स्टेट इज नॉन डैश डैश स्टेट। द नेक्स्ट, one should remember from physics that system at equilibrium cannot perform dash dash. The next, as living organisms work continuously, they cannot afford to reach dash dash. The next, hence the dash dash state is a non-equilibrium steady state to be able to perform dash dash. The next, hence the dash dash state is a non-equilibrium steady state to be able to perform work. Living process is a dash dash effort to prevent falling into equilibrium. The next, this achieved by dash dash input. The next, dash dash provides a mechanism for the production of energy. The next, hence the living state and dash dash are synonyms. The next, without dash dash there cannot be a living state. The next 9.7, yes 9.12 एंजाइम्स, जल्दी से रिवाइज कीजिए इसको। रिवाइज कर लिए। फर्स्ट क्वेश्चन, ऑलमोस्ट ऑल डैश डैश आर प्रोटीन्स। The next, there are some dash dash acid that behave like dash dash. The next, these are called dash dash. One can depict an enzyme by a line diagram. The next, an enzyme like any protein has a dash dash structure that is amino acid sequence of the protein. The next, an enzyme like any protein has dash dash and dash dash structure. The next, when you look at a dash dash structure figure 9.4b, you will notice that the dash dash of protein chain folds upon itself, the chain dash dash itself, and hence the dash dash or dash dash are made. The next, one such pocket, जी हाँ, one such pocket is the dash dash site. The next, an dash dash site of an enzyme is a dash dash or pocket into which the substrate fits. The next, thus enzymes through their dash dash site catalyze reaction at a high rate. The next, enzyme catalysts differ from inorganic catalysts in many ways, but one major dash dash need mention. The next, dash dash catalysts work efficiently at high temperatures and high pressures, while dash dash get damaged at high temperatures, say above 40 degrees Celsius. The next, however, enzymes isolated from dash dash who normally live under extremely high temperatures, example hot vents and sulfur springs, are stable and retain their dash power even at high temperatures up to 90 to 80 or 80 to 90 Celsius. The next stability is thus an important quality of such enzymes isolated from organisms. The next 9.12.1 chemical reactions. जल्दी से रिवाइज कीजिए इसको. चलिए the first question, how do we understand these enzymes? The next, let us first understand a chemical reaction. The next, dash compounds undergo two types of changes. The next, a physical change simply refers to the change in shape without dash of bonds. The next, this is a dash process. The next, another dash process is a change in a state of matter when ice melts into dash or when water becomes a dash. The next, these are also physical processes. However, when bonds are broken and bonds are formed during dash, this will be called a chemical reaction. For example, BaOH whole twice plus H2SO4 gives BaSO4 plus dash. The next, देखिए आपको साउंड क्वालिटी अच्छी नहीं आ रही है. हम इस बात को भली भांति समझते हैं क्योंकि यह जगह पूर्ण रूप से नेचर की गोद में है और यहाँ विभिन्न प्रकार के प्राणी अपना जीवन बसर करते हैं और सब अपनी लाइफ प्रोसेस चलाते हैं, इसलिए आवाज़ आना बिल्कुल संभव है. भविष्य में आवाज़ की गुणवत्ता सुधर जाएंगी. हम इस पर लगातार अपनी प्रयास कर रहे हैं. चलिए द नेक्स्ट क्वेश्चन, जो हम रिएक्शन आपको बताए हैं, बताइए वो कौन सा रिएक्शन है, इनऑर्गेनिक या ऑर्गेनिक. The next, similarly dash of starch into glucose is an organic chemical reaction. The next, rate of a dash or dash process refers to the amount of product formed per unit time. It can be expressed as dash. The next, rate can also be called dash if the direction is specified. The next, rates of dash and dash process are influenced by temperature among other factors. The next, a general rule of dash is that rate doubles or decreases by half for every dash degree Celsius change in either direction. The next, dash reactions proceed at dash higher than that of uncatalyzed ones. The next, when enzyme dash reaction are absorbed, the rate would be vastly higher than the same but dash reaction. For example, carbon dioxide plus water gives dash acid with dash anhydrase. The next, In the absence of any enzyme, this reaction is very slow, with about dash molecule of H2CO3 being formed in an hour. The next, however, by using the enzyme present within the cytoplasm called anhydrase, the reaction speed dramatically, with about molecules being formed every second. The next, the has accelerated the reaction rate by about millions time. The next, the power of is incredible indeed. There are thousands of types of enzymes, each a unique chemical or metabolic reaction. The next, a chemical reaction when each of the step is catalyzed by the same enzyme complex or different enzymes is called a pathway. For example, glucose to 2-pyruvic acid इसका फॉर्मूला बताइए. The next, a multi-step reaction when each of the step is catalyzed by the same enzyme complex or different enzymes is called a metabolic pathway. For example is, जो रिएक्शन दिया गया है उसको बोलिए. Is actually a pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. The next, when you study respiration in chapter you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two reaction give rise to a variety of end products. The next, in our muscle, under anaerobic condition, lactic acid is formed. Under condition, pyruvic acid is formed. The next, in yeast during the same pathway leads to the production of ethanol, that is alcohol. The next, hence in different conditions different products are possible. Thank you.
You are most welcome in the next session of this discussion series.